Hello all, welcome to Biology Knowledge World. So, in this channel, we will discuss about all the important topics related to the field of biology, which are useful for students, school, colleges, and also for competitive exams like CSIR, NET, GATE, etc. So, the videos of each of the lectures will come in the upcoming days. So stay tuned everyone and support my channel. Subscribe to my channel. Okay, let us start today's topic. So today we are going to take the class on the topic glycolysis. Okay, so I think glycolysis is one of the most important and easiest topics that everyone can catch up. Okay, so glycolysis is one of the most important and some of the exams it is one of the focus areas for almost every exams like CSIR NET and GATE. So for the upcoming GATE 2026, this will be one of the useful videos for you to easily understand the concepts and the topics. So, if you have any clarifications or about the GATE exams or any clarifications about CSR NET exams, kindly note down that in the comment section, and I will reply to every doubt that you guys are asking. So today we are going to discuss about glycolysis. So let us start. So glycolysis, what is glycolysis? Simply, glycolysis is the process by which glucose is converted to pyruvate. So this glucose can be converted into any molecule such as glycogen, that is a stored form or storage form of glucose, or it can be converted into ribose or NADPH, which enters to the pentose phosphate pathway. So now we are studying about the conversion of glucose to pyruvate. So glycolysis is one of the process which occurs in the cytosol or cytoplasm of a cell. It is considered as an anaerobic process because it does not require oxygen for all the steps. So let us go straight to the pathway of glycolysis. So, first of all, we know that glycolysis, the substrate of the process is what? Glucose. So, from glucose, the first product that is formed is glucose 6 phosphate. Okay, so what is glucose 6 phosphate? When the glucose gets a phosphate group at its sixth position, it is called glucose 6 phosphate. So the enzyme that helps in converting from or for the conversion of glucose to glucose 6 phosphate is hexose kinase. So always remember that kinase are certain type of enzymes that helps in the addition of phosphate group at a particular position. So always kinase enzymes use ATP as the energy molecule. So here the ATP is getting converted into ADP and inorganic phosphate. So from glucose 6-phosphate is formed with the help of hexose kinase enzyme. Here a molecule of ATP is utilized and it is getting converted into ADP. So the next step is from glucose 6-phosphate, we need to get the product that is fructose 6-phosphate. So fructose 6-phosphate, what is the what are the similarities between glucose and fructose? We have learned in this smaller classes that glucose and fructose are the isomerases, right? So from glucose 6-phosphate to fructose 6-phosphate, this conversion requires enzyme isomerase. And what is phosphoglucose? From the glucose, the glucose molecule containing the phosphate group is getting converted into fructose 6-phosphate, and hence the enzyme glucose 6-phosphate from the enzyme. This enzyme is called phosphoglucose isomerase. I think that is clear. Okay, so from glucose 6-phosphate is formed, and from glucose 6-phosphate, fructose 6-phosphate is formed. Always remember about the enzymes. We have learned about two enzymes right now, that is hexose kinase enzyme and phosphoglucose isomerase enzyme. Okay, the next step involves a conversion from fructose 6-phosphate to fructose 1,6-bis phosphate. What is the difference between fructose 6-phosphate and fructose 1,6-bisphosphate? We can see that in fructose 6-phosphate, the phosphate group is only at the 6th position, while in fructose 1,6-bisphosphate, the phosphate group or groups can be seen at 1st position and 6th position, right? The enzyme used here is phosphofructose kinase. So we have seen earlier that kinase enzymes are the enzymes which utilize ATP, right? So from ATP to ADP conversion is also involved here. Okay, the next step involves conversion from fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So the enzyme here involves, in this step, it involves the splitting of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzyme involved here is aldolase enzyme. So always remember that fructose 6-phosphate is a 6-carbon molecule which gets splits into 3 carbon molecules. That is, what are the 3 carbon molecules that are produced here? Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, so we can also see, just look at here, like the green colored enzyme that is written here, it is called triose phosphate isomerase. That is, both the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are 3 carbon, right? So these 3 carbon molecules are interconvertible forms. They are isomers of each other. So DHAP gets converted to glyceraldehyde 3-phosphate. The reason is that in the further down steps, only glyceraldehyde 3-phosphate can enter, but dihydroxyacetone phosphate cannot enter. So due to this drawback, DHAP can never enter into the next steps. This DHAP gets converted into glyceraldehyde 3-phosphate with the help of enzyme triose phosphate isomerase. I think that's clear. Okay. From glyceraldehyde phosphate, always remember that we already have one glyceraldehyde 3-phosphate, right? But from now on, the DHAP is also converted into glyceraldehyde. So we have an extra glyceraldehyde. So from now on, we are using 2 moles of glyceraldehyde 3-phosphate. From this step, from this to this step, we have seen that only 1 mole of substrates are used from, but now on we are using 2 moles. Always remember, from this step onwards, from these steps, we can see that 2 moles of the substrates are used. Okay, from glyceraldehyde 3-phosphate, let us check on for the next steps. From glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate is formed. The enzyme used here is glyceraldehyde 3-phosphate hydrogenase or dehydrogenase. Always remember that dehydrogenase enzymes are the enzymes that involves the utilization of NAD. NAD is another energy molecule that is used in almost all biochemical reactions, right? So from glyceraldehyde 3-phosphate, 1,3-bisphosphoglycerate is formed with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase. It involves the utilization of 2 NAD molecules. So how these 2 NAD molecules are used, or what is the purpose of using 2 NAD? The thing is that we have already told that there are 2 moles of glyceraldehyde 3-phosphate. Okay, so we need 2 energy molecule or 2 NAD plus molecules. So that is the purpose why we use 2 NAD plus molecules here. Okay, I think that's clear. Okay, from 1,3-bisphosphoglycerate, 3-phosphoglycerate is formed. The enzyme involved here is phosphoglycerate kinase. So in the earlier steps, we have already told that kinase. What is kinase? It involves the addition of a phosphate group, right? So it requires ADP. In these steps, from now on, ATP to ADP conversion cannot be seen, but instead ADP to ATP conversion can be seen. Okay, so from 1,3-bisphosphoglycerate, 3-phosphoglycerate is formed. Next step involves conversion of 3-phosphoglycerate to 2-phosphoglycerate. The enzyme involved here is phosphoglycerate mutase. Okay, from 3-phosphoglycerate, 2-phosphoglycerate is formed with the help of enzyme phosphoglycerate mutase. So from here the phosphoglycerate that was like 3-phosphoglycerate contains a phosphate group at the third position, while 2-phosphoglycerate contains phosphate group at the second position. Okay. Next, from 2-phosphoglycerate, phosphoenol pyruvate is formed. So it involves an enol form, right? So the enzyme involved here is enolase enzyme. Okay, from phosphoenol pyruvate molecules is formed. Here actually 2 moles of pyruvate is formed. The reason is that we use 2 moles of glyceraldehyde 3-phosphate, right? So the enzyme used here is pyruvate kinase. Pyruvate kinase. So we have already told that what kinase enzymes use ADP, right? So 2 ADP is converted into 2 ATP. So these are the steps of glycolysis. Let us just recall the first substrate or the first molecule involved in this step is glucose. From glucose 6-phosphate is formed. From glucose 6-phosphate, fructose 6-phosphate is formed. From fructose 6-phosphate, fructose 1,6-bisphosphate is formed. From fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are formed, but this dihydroxyacetone phosphate gets converted into glyceraldehyde phosphate. Then once this glyceraldehyde phosphate gets converted to 1,3-bisphosphoglycerate, then to 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and finally to pyruvate. So these are the steps of glycolysis. So basically this glycolysis is divided into 2 stages. The steps from here to here are called preparatory phase. It is called preparatory phase, and the steps from here to here, it is called payoff phase. Okay. The preparatory phase, it is preparing the glucose to enter to the processes of glycolysis. That is preparatory phase. Here, 1 mole of ATP, another mole of ATP. So a total of 2 moles of ATP is utilized here. But in the payoff phase, payoff means these 2 ATP, the 2 ATPs that are used in preparatory phase is given back because it is a taken one. So it should be paid back. That's why the steps from here to here is called payoff phase. Payoff phase. Okay, I think that's clear. So in payoff phase, 2 moles of NAD are formed, and here 2 moles ATP is formed, here 2 moles ATP is formed. That is 2 moles of NADVH is formed and 4 moles of ATP is formed. But in the first step or in the preparatory step, we have utilized 2 ATP, right? So 4 minus 2 is equal to what? 2. So the net ATP gain will be 2 ATP and the net NADH gain will be 2 NADH. So the bioenergetics or the energy gain in this process is what? 2 ATP and 2 NADH. So these are the steps of glycolysis and energy gain of glycolysis. So it is considered that glycolysis is an energy favorable, right? Because here the glucose is converted into pyruvate and 2 ATP and 2 NADH are also formed. So this is an energy favorable process and one of the most favored process that occurs in a cell. I think that the glycolysis is clear to everyone. If you have any doubts, just comment that down and I will reply to you. Okay, bye.